Okay, in this video I want to talk more about protein structures, and I don't have the question actually typed out or written out, so I'm just going to read it here. And it says, protein structures are usually presented as static figures, but the reality is that proteins are dynamic. Explain the chemistry behind the observations and suggest why this is beneficial to the organism. So they're really asking us about protein structure, and they say why it's usually presented as static, but in reality these proteins are dynamic. So why are they not static? Well, they're not static because proteins function often entails the interconversion between two or more structural forms. So let me write that down so that can get it on paper here. So it says proteins are not static because protein function often entails an interconversion between two or more structural forms. So what I said here so far is that proteins are not static because protein function often entails an interconversion between two or more structural forms. So proteins are not static because protein function often entails an interconversion between two or more structural forms. So let's see if we can relate that to something dealing directly with proteins, and more specifically enzymes. So the ability to change conformation. So the ability to change conformation is critical to protein function. So it's critical to protein function. For example, enzymes, which we haven't talked about yet, but we will. Enzymes often change conformation upon binding of the substrate. Now, you might be asking, well, he's saying this enzyme changes shape upon binding of the substrate, and this is not always true in all cases. Yes, you'd be correct. This is called the induced fit model. It basically states that enzymes change their shape to accommodate the substrate. So upon substrate binding, the shape of the enzyme will also change. And that's one example of proteins being dynamic. I mean, this thing is in motion. As soon as the substrate binds, the protein changes shape, and that's a dynamic feature. That's motion, you know, that we're talking about here. This movement, change from one conformation to the other. So basically, I would probably say this, and I would give that example, and I'd say proteins are not static because protein function often entails an interconversion between two or more structural forms. I say the ability to change conformation is critical to protein function. For example, enzymes often change conformation upon binding of the substrate. And, you know, just to show that I know what the hell I'm talking about, I might say something like, this is called the induced fit model. So I might just say that this is the induced fit model. Alright, so that's all I wanted to say about that, and hopefully this is helpful.